So our next speaker is Dr. Jessica Dominguez-Rieg from the University of South Florida, and she'll be talking to us today about intestinal epithelial function.
Good morning, everyone. Thanks for coming out today to learn about the GI system. Today I'll be talking about intestinal epithelial function. I'm going to give a brief overview of really just what the intestinal epithelium is and its barrier function, and then we're going to go through a few examples, predominantly about electrolyte and water absorption and secretion, just to give you a little bit of a refresher on that. So let's get started. As the name of this GI refresher course implies, the gut is not just the gut anymore. We're learning that that's actually a very elegant, beautiful system that incorporates not only the actual intestinal epithelium, but now we know that there is a whole other world living within our system and the microbiota, and that's what Dr. Collins will be talking to you about today, and also that the mucosal immune system plays a very essential role in helping to create the gut's function, and that's what Dr. Edelblum will be talking about today. And so for me, we are going to be really focusing on the intestinal epithelium itself. Now, the anatomy of the intestine, for those of you that aren't familiar with the way that the gut is composed, the epithelium is a constantly renewing, self-renewing source of epithelial cells. And the way that the intestine is laid out is like a carpet, where you have all of these villi that project out into the lumen of the intestine, and each of these villi are fed by numerous crypts, which reside down at the bottom, where there are stem cells that proliferate, and we have these proliferative crypts allow us to regenerate this constantly renewing epithelium. So as these cells are born down here in the crypts, several of them march up to the top before they're shed off at the top. And other cell types like the Panna cells will march their way down to the bottom of the crypt while they have their function down there. And so when we look at these villi crypt units, there's about 250 cells in each of these crypts and about 6 or 7 crypts that surround these individual villi. And this really is just a wonderful way that these cells can repopulate, and we continually shed our intestinal epithelium about every 3 to 5 days. Now, as this figure shows, there are stem cells located near the base of the crypt, and those stem cells give rise to really 4 essential types of mature epithelial cells within the intestine. We have the absorptive enterocytes, which are predominantly up here in the villi, and those are the cells that do most of the work of having the brush border membrane, having those digestive enzymes, as well as our ability to absorb the nutrients through those enterocytes. We also have mucus-secreting goblet cells because we need the mucus to help lubricate. And then we also have hormone-secreting enteroendocrine cells scattered throughout this epithelium that play a really important role in hormonal regulation of gut function. Those 3 cell types are the ones that march up the villi and eventually will get shed off. The 4th type of cell are these Paneth cells, and these are antimicrobial peptide-secreting cells that are very important for part of the immunological function of the intestinal epithelium here. Now, what's quite phenomenal, if you think about your intestinal epithelium, it is a simple columnar epithelium. There's one layer of cells there that's protecting all of the antigens and microbiota and any sort of pathogens that are out here in the lumen from getting inside your body. So when you think about the importance of the intestinal epithelium, it's absolutely critical. We just have this one little row of cells and an army behind it, which is the mucosal immune system, really preventing any sort of pathogens or toxins from being able to get inside our bodies, because ultimately we are like a big donut, and that everything on the inside of your intestine is actually on the outside of your body. The other important thing that I like to tell my med students, I teach the GI physiology in year 1. It's the very last course of their first year, so they're tired and they're ready for a break. And a lot of times they're not that excited about the gut, but GI physiology is extremely interesting and extremely relevant for students. And I always like to remind them that even Hippocrates said all disease starts in the bowel. So that this piece right here at this level of the intestinal epithelial barrier is super, super important and really can be the root of a lot of diseases that they'll learn about in later years. Okay, so that brings us to the actual intestinal epithelial barrier function. What's keeping the bad stuff that's on the outside from getting inside? And there's really three different components of this intestinal epithelial barrier. The first one being a microbial and chemical barrier. So in the lumen, kind of sitting on top of these villi and crypts here, there is a lot of mucus that the goblet cells have produced. There are commensal bacteria that live there, and you'll learn about the microbiota today. And there are antimicrobial peptides that are produced by the Paneth cells. There's also a lot of secretory IgA sitting there. So we already have several different chemical mechanisms just sitting there waiting to help us out and protect us from anything crossing this simple columnar epithelial layer. So that's one piece of it. And actually the details of how this mucus works is actually interesting because there's two different mucus types, and that really dictates where certain bacteria like to reside and how some of these chemicals actually diffuse through that mucus layer. But the mucus is important, more than just serving as a lubrication surface to move the chyme through the intestinal tract during digestion. The second piece of the intestinal epithelial barrier is a physical barrier, and that's really that single layer of epithelial cells themselves. These epithelial cells act like any other epithelial cells. They like to create very tight communication between each other, so the role of cell junctional protein complexes is absolutely critical for epithelial barrier function. And if you look at any sort of examples of problems with the intestinal epithelial barrier, we have to consider the role of the tight junctions, which are the ones that are going to really protect us from having bacteria or other things be able to squeeze through paracellularly into our bodies. So the tight junctions reside up here, up at the apical apical portion of these epithelial cells. They create a belt around the epithelial cell. So I like to think of the tight junctions like the plastic rings on a six-pack of soda or water, or whatever beverage of choice beer that you would like to think of, that the tight junctions really create a tight seal across that apical side that prevents things from leaking in. You also have additional cell junction proteins that are important for allowing the cells to remain in close contact with each other, both cell-cell junctions like the adherens junctions and the desmosomes, but also cell junction proteins on the basolateral side, which are allowing those epithelial cells to stay stuck down on the basement membrane and the submucosa down below it. But all of these junctional proteins can play an important role in pathology, but when we talk about barrier function, a lot of attention is really given to these tight junctions. And these tight junctions are composed of multiple proteins like claudins and occludins and zonula occludins that are really important. They all pair up differently, and depending on which type of claudin or occludin are expressed there, you get varying degrees of leakiness through that tight junction. And also, not only is it creating a cell-to-cell junction, but these tight junctions are also linked up to the cytoskeleton. They bind to F-actin, and so you can really use tight junctions as a great example of how things go wrong in inflammatory diseases that affect the leakiness of the intestinal epithelial barrier. And I think you'll hear more about that today in the next two talks. And then the third piece of the epithelial barrier is really the immunological barrier. So this is where the army comes into play. There's a whole army of immune cells that reside within the lamina propria of these villi and in the submucosa that are there to help protect us in the event that something actually crosses through that physical epithelial space. And this also involves a bunch of different cytokines, and I think Dr. Edelblum will give you a great overview of how this piece of the epithelial barrier works. And so when we're going through today in my examples of intestinal epithelial function, I mentioned some of the ways that we can affect the intestinal epithelial function by any sort of inflammatory process that ends up changing cytokine levels. And you'll see that as we go through the talk today. Okay, so overall, if you think about gastrointestinal functions, there's really 4 basic functions of the GI tract. Motility, so how you actually swallow, and all the way down to how that food is then moved through your intestine, all the way down to the anus. We have the second function, which is secretion. So in order to move things, we need lots of secretions, all the way from saliva in your mouth to the pancreatic secretions to the intestinal secretions. And we'll talk about this specifically today. In those secretions, we have lots of pancreatic enzymes, as well as buffers like bicarbonate. So that allows us to have appropriate digestion of the food. And then once the food is properly digested, then we have the 4th function, which is absorption. And absorption will take place across those epithelial cells through very particular processes and transporters, which allow us to bring the nutrients into the bloodstream. So what I wanted to do today is really focus on these 2 epithelial functions, which is secretion and absorption. And we can talk about lots of different things that involve secretion and absorption in the intestine, but I chose to focus predominantly on electrolyte and water, excuse me, secretion and absorption, and how that works within this intestinal epithelium. Now, classically, it's taught that secretion predominantly takes place in the crypt compartment, that these cells have more of a secretory role in secreting electrolytes and therefore water out into this lumen space of your intestine. While the absorption piece is predominantly taught that takes place in the villus compartment, and that's where your nutrients would be digested and then absorbed across these enterocytes. So let's look at how that works a little bit more closely. One important thing, if you look just at the basic anatomy of these crypt villus compartments, there is a lot more villi compared to crypts in terms of surface area. The other thing that's important is if you just step back and think about the actual anatomy of the intestine, it's actually organized into several different layers, all designed to help maximize surface area for absorption of the nutrients. And so if you think about this tube, it's not just a simple cylinder that lives within our intestines. It's actually a cylinder that's thrown up into folds. Those folds then have several different villi projecting out into them, and on top of every single epithelial cell, every one of those enterocytes in the villi, you have microvilli, and that's the brush border membrane where a lot of those digestive enzymes occur. So as we move from a cylinder to throwing it up into folds, to villi, to microvilli, we're actually able to significantly increase the surface area that's available for absorption of nutrients. Now, previously it was thought that if you were to take all of this epithelium and smush it out into a single square, that the surface area is about 300 square meters, which is the size of a tennis court, which is really a 600-fold amplification from a simple cylinder to how it's really structured with villi and microvilli. That's a lot of surface area that's available for things to happen within the intestine. Now, with the development of better tools to actually measure the actual surface area within the intestine, it's been revised down a bit by about 10-fold. And then now the new estimate is that if you were to flatten out your intestinal epithelium, it's about the size of half of a badminton court. So that's still quite a lot of surface area if you think about our general body and how this intestine is wrapped up inside your abdomen, and that even half of a badminton court is a lot of surface area available for absorption of nutrients. So let's talk specifically about water and electrolyte secretion and absorption. So like I mentioned, if you look just at the general number of cells and the actual surface area of the villi versus the crypts, there's a lot more surface area up in the villi, and because of that, the role of the villi becomes more predominant. And so by far, if you think about secretion and absorption of nutrients, in the villi, where we have mostly an absorptive phenotype for absorbing things like sodium, chloride, and water, versus the crypt compartment, where these crypt cells are important for secretion of sodium, chloride, and water, then because there's a lot more surface area of the villi compartment, that we understand that water absorption is predominantly favored. So there's a lot more absorption of water and electrolytes compared to secretion, but they're both playing an important role in that GI function. So if you just look at how fluids are handled within the gastrointestinal system, and think about all the secretions that are added throughout the process of digestion, we have fluid that is entered from dietary sources, whether within our food or drinking, and our mouths, the salivary glands contribute another 1.5 liters of saliva each day. You mix that with all of the secretions that come from the stomach, which is another 2 liters. We're talking already by the time that the food enters the small intestine, we have a lot of liquid there. In addition, we have secretions from the gallbladder. We've got bile coming out. We've got pancreatic secretions. The intestinal epithelial cells themselves are also contributing secretions from that crypt compartment. So by the end of the day, by the time we get to the colon, we're really absorbing about 8.5 liters of fluid. So that by the time any of this chyme and secretions reaches the colon, we're really just fine-tuning that final absorption of water, which is in about another 400 mils, so that in the stool, we're really only excreting about 100 mils of water. So we have all of these secretions, about 9 liters of secretions that enter the system, and we're reabsorbing almost all of it, except for this 100 mils. So there's a massive absorption. I just taught kidney, so I'm in kidney mode right now. Lots of absorption happening of that water. Okay, and along with that, what we have to consider is not only how the water gets absorbed or secreted, but what is going along with that water. And that's really where we talk not only about absorption of nutrients, but also all the electrolytes. And we can talk about lots of different types of electrolytes that are absorbed across the intestinal wall, but I chose to really focus on 2 predominant ones that dictate absorption and secretion. One being sodium, which is really important for absorption, and the other being chloride, which is the major component for the secretory capacity of those crypt cells. So we're going to look at specific transporters and the key players that are really involved with handling sodium, chloride, and therefore water because of the osmosis. Okay, so here are some of the key players that are involved with electrolyte absorption and secretion. If we consider what's going on with absorption, there are several transporters present on both the apical and the basolateral membrane of those epithelial cells that allow us to bring in sodium and then have paracellular movement of chloride and water that follows. And so there are, in the proximal part of the small intestine, sodium nutrient transporters. So there's sodium-dependent glucose uptake, there's sodium-dependent amino acid transporters. And so we'll look at some of those. Another very important key player is sodium-hydrogen exchangers, which is also present on the apical side of the cell. These can functionally be coupled to these chloride bicarbonate transporters, and I'll show you what that looks like. Later on in the colon, we have the epithelial sodium channel, and I'll talk about that more specifically. And then, of course, all of these sodium gradients are driven by the sodium-potassium pump, which is present on the basolateral side of the cell. Very important to ensure that this gradient for sodium to come from the lumen into the bloodstream is present. And then in terms of the key players which are involved for secretion, we'll talk about the chloride channel. So we just heard in the previous talk about cystic fibrosis, so we'll talk about the CFTR chloride channel that's involved with that and how that plays an important role in secretion. And also the sodium-potassium 2 chloride transporter, which is present on the basolateral side of the cells, which allow all of those electrolytes to move into the cell and therefore secrete this chloride out, and then you get movement of sodium and water out of the intestinal epithelium into the lumen. So how does this look in terms of what these cells actually look like? And I like to have my med students draw lots of boxes with cells so they understand where the transporters are, which side of the cell they're on, so they really can follow through how an ion gets moved from one side to the other and what's actually going on there. So this is a simplified intestinal epithelial cell that's not region specific, because there are different expression of these transporters depending whether we're talking about the duodenum, jejunum, or ileum, or colon even. But these are the predominant key players that I just mentioned in the previous slide. So here we have the sodium-potassium pump on the basolateral cell, really providing the gradient to move all of these things through these channels that are and transporters that are present on the apical side. And as I mentioned, here up in the proximal part of the small intestine, we really rely on sodium absorption through these sodium nutrient transporters. So here you can see both sodium glucose and sodium amino acid transporters that allow those to come into the cell. And then the sodium would leave through the sodium-potassium pump, and either the glucose or amino acids would leave through an additional transporter on the basolateral side to enter the bloodstream. But for purposes of simplicity, I kind of tried to simplify and only really look at the sodium pieces here. In addition, we have the sodium-hydrogen exchanger. There's two predominant isoforms that are expressed on the apical side of the intestinal epithelium in the gut, but it's really NHE3 that seems to play the predominant role in the sodium absorption in these cells. And this process can be functionally coupled with this transporter, which is a chloride bicarbonate exchanger, which has varied expression depending on where we are. And I have a slide for this when I'll show you in more detail in a few minutes. And then later in the colon, we have this epithelial sodium channel, or ENaC, which plays a really important role in electrogenic absorption of the sodium. And then once we move the sodium across, then you can see that there'll be a gradient for chloride and water to follow through the pericellular route, so that we're really getting that final absorption of salt and water. And there, of course, are many other ion transporters that are present in these cells that we could talk about. I'm not able to cover what's going on, for example, with calcium or potassium. So it's important to remember that there's many processes happening here. This is just a kind of snapshot for sodium absorption in these enterocytes. Okay, so let's whet your appetite a little bit and talk about some of the relative things that are really we're starting to understand more about these individual transporters. And so the first one that we can talk about is one of the sodium nutrient transporters, for example, the sodium glucose transporter, SGLT1. So this is the way that we bring glucose into the cell, and it is sodium dependent, and then the glucose can go out through GLUT2 on the basolateral membrane. The sodium gets extruded through the sodium-potassium pump. And over the years, we've learned a lot about the function of SGLT1, not only just for being able to absorb glucose and sodium, but also what are the consequences if something goes wrong with this particular cotransporter. Both in humans, we know that in humans that have a problem with SGLT1, they end up not being able to bring this in, and you can end up with a congenital diarrhea, which we'll talk about later when we talk about some of the mechanisms of diarrhea. But we do know that even in both humans and in animal experiments, if you knock this transporter out, so these SGLT1 knockout mice, they have a whole host of problems, but one thing that they exhibit is severe osmotic diarrhea. And so then we like to think through, why would that be the case? Why would these mice have, or even humans, have an osmotic diarrhea because they can't bring in sodium or glucose? And then we can talk about how, well, if it can't get in, then there's a lot more sodium and glucose left in the lumen, which has an osmotic capability to hold the water in the lumen, and so you end up with diarrhea. So there you're really fighting against the drive to absorb these salt and water, electrolytes and water, across those cells. In addition, we know that there is actually a much more elegant regulation of this SGLT1 transporter, and there's many things going on as we're learning about this, but one thing that I think is really interesting is how other GI hormones can help actually regulate this transporter, and we don't know all the answers to this yet, but we do know that this SGLT1 expression, that transporter can get taken out of the membrane and shuttled back to the membrane for a whole host of reasons. One of them seems to be that in mice that are hyperleptinemic, so they have very high circulating leptin levels. This is often associated with obesity and type 2 diabetes, that if there is a presence of high leptin there, that SGLT1 is taken off the apical membrane. And if it's not there on the membrane, then we have to rely on other transporters and other ways to get glucose and sodium in. And so that's one of the examples that we like to talk about in class is, we learn kind of classically in the textbooks about SGLT1, but what are some of the new things that are coming out as we learn about this particular co-transporter? In addition, you can't really watch TV these days without seeing some sort of commercial for an SGLT1 or 2 inhibitor for the treatment of diabetes. And so I think this is a very relevant example to talk to students about because they see it on TV every day. And maybe they haven't had the pharmacology behind these drugs yet, but they should be familiar with the fact, if they watch those TV ads carefully, that there is a lot of pharmacological targeting to SGLT1 and 2, which is present in both the intestinal epithelium as well as in the kidney. Now, a lot of those drugs were targeted in the kidney so that if you block reabsorption of the glucose, that you pee out the glucose, which would be good for treating diabetes. But the recently developed a drug called sotagliflozin, which is specific. It's an oral version of an SGLT inhibitor that is designed to block SGLT1 in the intestine. So really going at the other end, what happens if you treat these patients by blocking glucose entry from the very beginning? And there's been a few clinical studies kind of showing that this might be an effective treatment for diabetes. So really just preventing glucose from getting in the first place. And what's interesting to try to postulate is, okay, that's great. This is another good target for treating diabetes, but how does this tie in with studies that show that, for example, in hyperleptinemia, that SGLT1 is not even in the membrane. So can that drug do something if SGLT1 isn't even present there? And so this is just ways to kind of get students to think and process through the basic information and try to apply that to things that they will come into play with as they see patients in their future years, as these drugs continue to be introduced to the clinic. Okay, so what about NHE3? NHE3 is that sodium hydrogen exchanger where we bring in a sodium and extrude a proton. So as you can already tell, this really has two important functions. Number one, to bring in the sodium. Number two, this is extruding a proton, and because we're extruding a proton, then we have to also talk about the importance of the intestine with acid-base balance. And so we explain how within these epithelial cells you have carbonic anhydrase, which is the enzyme that helps produce carbonic acid, which can then dissociate into a proton and a bicarbonate. And it's that really supplies the proton to be extruded. And that's why we talk about this functional coupling with this DRA or PAT1, this chloride bicarb exchanger that allows us to extrude a bicarb and a proton out into the lumen of the intestine while bringing in a sodium and a chloride for absorption. And so it's really important to think about some of the things they're learning about acid base as well, and how that plays a role into feeding into all these transport mechanisms. We also know NHE3 is extremely important and relevant in inflammatory diseases of the gut. So if you knock out this exchanger in mice, they exhibit severe diarrhea. They have metabolic acidosis because you can't actually get this proton out. And they have increased mortality. They don't really live very long because of these severe problems. And some of the pH that you see in these mice, it's pretty significant that they make it that long at all. But this can cause a lot of problems in mice that don't have this particular sodium hydrogen exchanger. In addition, we know that loss or inhibition of NHE3 is associated with lots of other things that you might imagine, having problems with the pH in the lumen, for example, that this has been associated with gut microbial dysbiosis. If you change the pH, perhaps different bacteria are then allowed to grow, and so you can really change the microbiota and therefore the consequences of those microbiota if you mess with the pH, and you can mess with the pH by having issues with the sodium-hydrogen exchanger. Because of that, you might have an epithelial barrier defect and inflammation that happens in response to the lack of this NH2/3. And so we know that if you look across different types of inflammatory bowel disease like ulcerative colitis and Crohn's, a lot of these patients tend to have reduced expression of NH2/3. And so that's one of the targets of thinking about how to treat patients, is how to handle the fact that this might be decreased in expression. And NH2/3 is actually regulated on multiple different levels, which we could spend a lot of time talking about, but I'll let you look at that on your own. But this transporter is regulated and shuttled back and forth onto the membrane as well, so there's lots of things that can affect whether NH2/3 is present on that apical membrane or not. But it's something that's very important for sodium absorption as well as the acid-base piece of what the intestinal epithelium is responsible for. In addition, there has been some interesting studies that have come out recently. One of the pharmacological targets that actually has a cardiovascular impact was the development of tenapanor. Tenapanor is an NH2/3 inhibitor. It's an oral NH2/3 inhibitor because we have NH2/3 in other places like the kidney. So this was to specifically block NH2/3 within the intestinal epithelium. And what they actually started giving tenapanor for was to decrease hyperphosphatemia in patients. And for some unknown mechanism, when you block NH2/3, you affect phosphate absorption through those enterocytes. And so that was one kind of off target of this drug was, hey, we can really decrease cardiovascular risk in patients that have high phosphate levels by inhibiting a sodium-hydrogen exchanger. And so there's a lot of research currently going on to understand what is the actual mechanism involved here. However, there was a recent paper that came out, a clinical study that was looking at what happens in patients with hyperphosphatemia if you block this sodium phosphate transporter, which is isoform 2B in the intestinal epithelium. Because one suggestion for the NH2/3, the effect of blocking NH2/3 was perhaps it inhibited NaPi2b. But when you actually jump the gun a little bit and inhibit NaPi2b specifically, it actually had no effect on their phosphate levels whatsoever. So they followed patients over several days, and they looked at fecal phosphate excretion, and it didn't really matter whether the patients received this inhibitor or not. Their phosphate levels didn't go down. So this is really suggesting that perhaps the mechanism of a drug like tenapanor, which is inhibiting NH2/3, isn't really treating hyperphosphatemia by inhibiting this other sodium phosphate transporter, NaPi2b. So this is stuff that's going to be coming out soon to try to really understand the relationship between a sodium hydrogen exchanger and a sodium phosphate co-transporter. And so I think this is something to look out for because it's very relevant to medical students to see what these drugs are doing at a basic science level that they're seeing in patients. And obviously cardiovascular health is something that's very, very important. It's one of our top killers in the United States. And so this is just really showing them, again, why the gut is really important for something like cardiovascular health. Okay, so then I mentioned that NH2/3 can be functionally coupled to this chloride bicarbonate exchanger. And so just to give you a little bit of information about that, this DRA or PAT1 can be expressed in different regions. DRA is predominantly expressed in the duodenum and colon, and PAT1 is predominantly expressed in the small intestine. And so a little bit varied degree of expression. And then as I already kind of described, that you have this functional coupling between sodium and chloride reabsorption and then bicarb and proton secretion out of those epithelial cells. And we know that by knocking out those transporters, we can see the effects of diarrhea. So for example, DRA knockout mice exhibit severe diarrhea and loss of chloride in the stools, so that's a chloride-rich diarrhea. However, if you knock PAT1 out, we don't really see the same effect. So they're not really the same exact function or the transporter, and in fact, PAT1 is an anion exchanger that can exchange many more things besides bicarbonate, and so perhaps that's playing a different role. But again, the important piece to take home is really that you have this functional coupling for absorption of electrolytes between these two transporters. Okay, what about the colon? So in the colon, the predominant way that we absorb sodium is through the epithelial sodium channel, or ENaC. This is a heterotrimeric channel for electrogenic absorption. The previous transporters I mentioned are for electroneutral sodium absorption. This one is electrogenic. And the way that this channel is regulated is very similar to ENaC in the kidney. And by that, I mean that this channel can be upregulated by aldosterone and glucocorticoids, just like we see in the kidney. So just like we talk about the aldosterone-sensitive distal tubule, we also have an aldosterone-sensitive colon. And that is really important for really the fine-tuning of sodium absorption within the colon. And we also know that in diseases like inflammatory bowel disease, there's reduced expression of this epithelial sodium channel ENaC. And so these are things that people have studied and tried to understand why there is reduced expression of some of these transporters. And we'll talk a little bit about that here at the end. Now, there are also important key players for chloride secretion. This would be coming from the crypt compartment. And by that, we're really talking about two types of chloride channels that live on this apical surface, one being the CFTR channel, which is the cystic fibrosis channel for chloride secretion, as well as a calcium-activated chloride channel. There's also this basolateral sodium-potassium 2 chloride co-transporter. It happens to be isoform 1. That is really the loader to bring in these electrolytes into the cell so that the chloride can be secreted. Of course, we have the sodium-potassium pump, which is important for maintaining these gradients. And because we know that CFTR has the capability to also secrete bicarbonate, there has to be a way for bicarbonate to get into the cell, so there has to be some sort of anion exchanger present on the basolateral side as well. But the sum would be that you have chloride secretion, and then there is a drive for sodium and water to move through paracellularly into the lumen. Also, in terms of secretion, there's another route to induce secretion besides just regular ion-dependent secretion, and that is more of the neuroendocrine side, where you have things such as acetylcholine or VIP or histamine being able to bind to the basolateral side of those cells and induce chloride secretion through those channels. Okay, so let's look a little bit at CFTR. CFTR is important for regulated secretion of chloride and bicarb via a cyclic AMP-PKA pathway. And so the adenylyl cyclase second messenger signaling system is very important for how this particular chloride channel works, which is why we use it as a classic example of enterotoxin-induced secretory diarrhea like cholera. And as mentioned in the previous talk, by the time med students get through their first year or so, they've heard about cystic fibrosis and cholera a lot. And so they're very familiar with the fact that, okay, cholera causes upregulation of the CFTR channel, and that's why you get a secretory diarrhea. And so we've really worked to try to spice that up a bit. And at the end, I'll give you one of our active learning examples of how we can really take that mechanism of the secretory diarrhea and make it very relevant within integration of the entire body and what's actually going on with a patient with cholera. And we'll talk about that at the end. In addition, we have this calcium-activated chloride channel. This particular chloride channel seems to be really important for viral-induced diarrhea. And so, for example, rotavirus is able to change the intracellular calcium concentrations, and by increasing calcium concentrations, it actually increases chloride secretion through this channel. And so we have really the effects of both bacteria and viral induced secretory secretion, secretory diarrhea by increasing chloride movement through those two channels on the apical membrane. Now, I mentioned that this has these are classically taught in textbooks and in most schools these days, that the villus compartment is responsible for absorption and the crypt compartment is important for secretion. But there has been several challenges to this model throughout the years, and Walter Boron's group has really just used some new tools to really better understand if that's really the case or not. And so if you think about what happens whether the cell has differentiated into a crypt cell that's having a secretory function or a villus cell that has an absorptive function, the machinery that's on them has to change, right? And so if we are moving from a crypt cell and marching up and becoming a villus cell, we have to change the transporters that are present there in order to move from secreting chloride to actually absorbing the sodium. And so is the sodium absorption really strictly limited to the villus absorptive cells, and does anion secretion only happen in the crypts? And so if just based on what we've reviewed today, what would need to happen is that as these cells differentiate from the crypt to the villus, they have to turn off anion secretion. So they have to figure out a way to decrease NKCC1, which is that sodium potassium chloride transporter. They have to get rid of CFTR and the calcium activated chloride channel, and instead initiate sodium chloride absorption, so then pop up SGLT1 or NHE3 or DRA onto that apical membrane to then flip the switch to then bring in the sodium instead of secreting the chloride. This takes a lot of effort for the cells, if you think about it. There needs to be a lot of energy and machinery in order to turn things off and put things on. So is this really how it works? And if you've looked around at GI research and other fields over the past few years, we've seen this huge rise in the use of enteroids and organoids in research to really study how these organs work. And so specific to the intestinal epithelium, there is a way to grow these intestinal enteroids, which are really harvested intestinal crypts that contain the adult stem cells that are then grown with particular nutrients and media to develop into these intestinal enteroids that allow us to study lots of different functions. And because they're derived from the intestinal stem cells, we have the ability to have an undifferentiated phenotype, depending on what growth factors are in the media, that are much more crypt-like, that would therefore be more like the Paneth cells and those transit amplifying cells. Or we can change and really push them to differentiate and become much more villus-like, where we would have the enterocytes, goblet cells, and enteroendocrine cells. In addition, if you take a pluripotent stem cell and differentiate that, that's the difference between an organoid and an enteroid. The enteroid is really just specific to the adult stem cells that are located in that crypt region, whereas the intestinal organoid is really coming from embryonic stem cells. But these intestinal enteroids have really given us a new way of looking at how those cells function, and they end up looking like little mini guts with villi and crypts in them. And that's really important for studying not only just basic physiology of how we think things work, but also using them to look at additional things like host pathogen interactions and drug screenings. But Walter Boron's group did a nice job of using these intestinal enteroids to really look at transport within these undifferentiated crypt-like cells and the differentiated villus-like cells. And what they're now proposing is that perhaps you don't have these cells so set in stone that one is absorptive and one is secretory, because when they look at these enteroids, these undifferentiated cells that live in the crypts actually express all of the proteins and transporters that we talked about. Not only do they have the chloride channels like we would expect them to have for a secretory cell, but they also express NHE3 and DRA as well. And so that gives us an important way of thinking about this, that perhaps it's really just as they differentiate, something gets expressed more, but they still have the ability to secrete anions and also absorb the sodium. So in an undifferentiated crypt cell, the predominant expression and the predominant role is still to secrete chloride. But as we actually differentiate up into the villus, then there is actually upregulation of NHE3 and DRA1, which would then switch the phenotype to being much more absorptive in nature. And so I think that's really an interesting way to talk to students about, okay, these are the details, but is that how it really works? Would that make sense for how the body actually puts things together? And so this is something that's new, and I think it's just a good example to show how with the latest developments of research tools, we actually go back and look at how physiology was originally studied and kind of see if that still holds true or if maybe there's a new way of thinking about it. Okay, and so the last thing I want to talk about is really how we engage students in learning about intestinal transport. A lot of times when you talk about this sodium transport, this chloride transporter, I start to gloss over and they're like, I don't really care, that's not relevant to me. I would rather talk about something more clinical. Well, there's lots of ways that you can integrate intestinal transport into clinical application for students. And I think that's really one of the exciting things about the gut is that it's actually very easy to come up with relevant ideas to get them excited and get them engaged in learning about how the basic physiology works in the intestine. So one classic example is just talking about the different types of diarrhea. There's many mechanisms of diarrhea. We can talk about congenital diarrhea, we can talk about bacterial and viral diarrhea, we can talk about inflammatory diarrhea. All of them have a little bit different way of actually causing something like a secretory or osmotic diarrhea. And then really the last thing I'll do is give you an example of how we try to even take that further and integrate that within the whole body to get them really thinking and trying to integrate not only their GI physiology, but also things like the cardiovascular system. And so we can talk about different diarrheas. Once they know how these transporters work and where they're located, then it's easy to kind of explain to them, okay, well, what happens when things go wrong? When you have a congenital diarrhea, what does that actually mean? Well, you, for one, might not have SGLT1 expression, which would cause a glucose-galactose malabsorption. That means you have more glucose and galactose in the lumen that's going to cause an osmotic diarrhea. So that's one example. If you end up with a mutation in this chloride bicarbonate exchanger, then you would have a congenital chloride diarrhea because the chloride can't get absorbed. You end up with very chloride-rich diarrhea. And again, an osmosis pull to keep the water in the lumen. Similarly, you can have a congenital sodium diarrhea. This has been linked with mutations in NHE3 and not being able to have that sodium proton exchanger there. And again, if you can't bring the sodium in, there's more sodium in the lumen. There's that osmotic pull to keep the water there. So that's kind of just the simple way of thinking about what happens if you don't have those transporters. But we can also look at bacterial and viral diarrhea. And depending on whether we're talking about bacteria like cholera or viruses like rotavirus, there's different mechanisms. And one thing that is important to realize is there's lots of intracellular messengers that really regulate expression of these channels, as well as the transporters in these more absorptive type cells. So for example, things like cholera toxin or toxins from E. coli or viruses like rotavirus can cause ultimately an increase in something like cyclic AMP, or if it's rotavirus, we're actually increasing intracellular calcium. That high level of those intracellular second messengers can, can then actually stimulate chloride secretion through the calcium-activated chloride channel if we're talking about rotavirus, or through CFTR if we're talking about cholera toxin and other types of enterotoxins. And because you get this upregulation of chloride secretion, then you end up with that secretory diarrhea. In addition, we have stimulation. For example, cholera toxin is thought to perhaps increase VIP, which then again would stimulate more cyclic AMP to then drive that chloride transporter. Further, we can have invasive bacterial species that make it into the intestinal epithelium, which gets the army of immune cells all up and running. So there would be a massive increase in cytokines, and the cytokines themselves can also contribute to increasing these second messenger signaling cascades to increase chloride secretion. One reason for that might be is to flush out these bacteria. We want to try to flush out the initiating cause there. Also, cytokines have the ability to interact with those tight junction proteins and can actually open them up and make them more leaky. Further, these second messenger signaling things like calcium and cyclic AMP are not only in these more secretory-like cells, but they're also in the absorptive-like cells, and we know that high levels of those actually inhibit things like NHE3 and ENaC, which is why we probably see a lot of downregulation in inflammatory bowel disease. And so this really is giving us both a decreased sodium absorption and an increased chloride secretion in terms of secretory diarrhea. Now, inflammatory diarrhea would be a little bit different. This is where we're really going to depend on the commensal bacteria and the presence of these toll-like receptors, but that can initiate signaling events like activation of NF-kappa B and production of cytokines. Again, high levels of cytokines can also influence things like the tight junction protein. They also might directly interact with the mucosal immune system to increase cytokines. Those can increase intracellular calcium and things like cyclic AMP or cyclic GMP to again drive chloride secretion out of those channels, or mess with the tight junction proteins and make them more leaky. And also, again, just like we saw in the previous slide, an increase in these second messengers can inhibit these sodium absorption mechanisms as well. So again, you get a decreased sodium absorption and an increased chloride secretion. So those are just ways to kind of tie in, hey, everybody can relate to diarrhea. We talk about different types of diarrhea, what's actually going on with the transporters that are really dictating electrolyte and water absorption and secretion. Now, one thing that I found to be really successful with our students in terms of getting them to have fun and appreciate and really think about transport processes is to use a clinical vignette and have them think through not just what's going on in the GI tract, but what's going on in the entire body of a patient. And so this is just one example where we would have a 38-year-old woman with a 5-hour history of profuse, watery, non-bloody diarrhea. It's non-odorous. She's having severe leg cramps. Her blood pressure is low. She's got a blood pressure of 85/50. Her pulse was 150 beats per minute. Respiratory rate is 30, so she's not feeling very good. The patient is lethargic but afebrile. She has minimal abdominal findings. She's got splotchy cyanosis. Her feet are cold. She has a barely palpable pulse in those extremities. And initial lab values indicated elevated white blood cell count. She's got an elevated hemoglobin. Her sodium levels are actually within normal range, but potassium is very low. And in terms of her acid-base status, her bicarb levels are also low. And so then 2 hours after admission, her bicarb level dropped even further to 5 milliequivalents per liter. And so we're suspecting cholera, which is what actually she ends up having happen. But instead of just the cholera piece on what it's doing to cause the diarrhea, we have them think through how that's actually going to affect the entire body, particularly the cardiovascular system. So once they kind of have the background for how cells either absorb or secrete fluid, then they understand that if there's a balance between absorption and secretion, that you end up with normal stool consistency. But with our patient, she has profuse watery diarrhea, so in that case, they are able to think through, well, if there is increased water over here, then there's probably a lot more fluid secretion than fluid absorption. And that is really, in this case, happening because of the effects of cholera toxin. And so cholera toxin, we know the mechanism is to eventually increase cyclic AMP. That increase in cyclic AMP drives chloride conduction through the CFTR channel, as well as the cyclic AMP-sensitive potassium channel, which comes into play with some of her other symptoms, so that you get this chloride secretion, which is keeping the water there. You end up with increased sodium and water movement paracellularly, ultimately leading to that profuse watery diarrhea. And then also we know that an increase in cyclic AMP is also able to inhibit these transporters that are important for sodium absorption. And so we really have kind of two things happening at once, which is causing this elevation of fluid secretion and a decrease in fluid absorption, leading to the profuse secretory diarrhea. And then what I do is have them think through the entire thing, what's going on here. So we start with, okay, she has profuse secretory diarrhea. What is that going to do to her body? Well, she's losing a lot of fluid, so she's probably going to have some dehydration. And we talk about the fact that secretory diarrhea is an isotonic dehydration. It's isotonic diarrhea. You lose just as many solutes in water as you would in the plasma. But still, the loss of fluid can cause hypovolemia. It can cause a drop in her blood pressure. It can cause this feeble or rapid pulse, an increase in heart rate. It can have events in the kidney by decreasing renal blood flow. And so they start to be able to build, okay, I have diarrhea in my GI tract, but it's affecting my volume status, and so it's going to have effects on my kidneys and my blood pressure. And then also, in addition, because of the loss of volume of fluids, then we also have decreased tissue perfusion, which can have effects like switching things to building up more lactic acid, which can have effects on her acid-base status. We also know that this profuse secretory diarrhea will be associated with bicarbonate loss in the stool. And so because of that, they understand that when you lose too much base, you can end up with metabolic acidosis, but there's other things that are factoring into there. For example, the fact that there's lactic acid building up can also contribute to the acidosis, as well as changes at the level of the kidney, when the kidney might not be handling the protons and bicarbonate as much as they were before as well, that we can end up with multiple reasons for having metabolic acidosis. And then they then say, okay, what do I do about metabolic acidosis? Well, then I'm going to have some sort of compensatory respiratory alkalosis. And so they understand really the consequence of losing bicarb in the stool, how that has effects in the entire body in terms of acid-base status, and also how we can shift these protons, swap them with potassium in order to help decrease that acid load in the extracellular fluid space. And then in addition to losing bicarb in the stool, they're also losing potassium in the stool because of that cyclic AMP dependent potassium channel, and that can have consequences as well. And this hydrogen potassium exchange would also be affected in that case. And so what they end up doing is kind of building out a flow chart for how something like diarrhea can have entire an integrated body response. And then they understand why the patient has the values that she has. Well, she's got normal sodium levels, which is why she still has isotonic dehydration, but she's dehydrated, so her hemoglobin levels come back up. So they're really able to start integrating all the things that they've learned so far in the year to really put this into perspective in a clinical way. Her blood pressure is low. They have evidence of that. Her blood pressure was given at 85/50, and also her tachycardia was also given. It's 150 beats per minute. So they understand, well, those are consequences of being dehydrated from the diarrhea. So they're able to link everything back up. In addition, the BUN levels are elevated, which is an indication that there's decreased renal blood flow to the kidney. We know that she has splotchy cyanosis, and she's got cold extremities, cold feet, which is a sign of decreased tissue perfusion in those extremities, and so that makes sense because of the dehydration. We also had two values for her bicarb, one being that it was low already, but then dropped to be even lower. And so they understand that the bicarb levels in the plasma will be low if she's losing bicarb in the stool. And so they understand how that happens. And then they should have already had acid-base regulation, so they understand how this metabolic acidosis and respiratory alkalosis work, which is why she has an increase in her respiratory rate, because she's trying to blow off the CO2 from this acidosis. So they are actually able to put a bunch of different things together just on a cholera case with profuse secretory diarrhea. And then the effects of potassium, in part, can be due to why she has leg cramps, in addition to how acidosis changes the ionic calcium values. But this is just a neat way to really think about how a GI dysfunction of the intestinal epithelium, like cholera, which they actually, at least in our curriculum, are extremely comfortable with by the time that they get to our course at the end of year 1. This really just allows them to take that information and build it out onto what they've learned throughout the rest of the year in an integrated way. And really trying to help them understand normal physiology versus disordered physiology and training their brains to think about the pathophysiology that they'll get later on in year 2. And then we can talk about different therapies. For example, if we have an increase in fluid secretion, then how can we inhibit these mechanisms to decrease the secretion, or how can we stimulate sodium reabsorption to then prevent this inhibition that's happening because of the increase in cyclic AMP. And they're classically, one of the classic examples when you talk about cholera is the development of oral rehydration therapy. But I think it's important for the students to realize why that works and why something like isotonic saline doesn't work as well. And so I show them this, how if you just give isotonic saline, you're not really stimulating more sodium reabsorption. So you still end up with more sodium in the lumen, which still means you have more luminal fluid volume. So you still have the secretory diarrhea. But if there's a way to kind of trick the system into reabsorbing more sodium by using something like the sodium glucose transporter, then we actually can stimulate sodium reabsorption, help mop up some of that extra fluid in the lumen, and also by adding glucose to the solution. And that's really why oral rehydration therapy works so well in the case of cholera. There's also a lot more than just that. There seems to be a secondary effect that by giving this oral rehydration therapy and stimulating SGLT1, it actually blocks the inhibition on NHE3, so you kind of get a win-win there to increase sodium absorption. And in addition, because of the case where we talked about additional things like her acid-base status and the loss of potassium, we talk about oral rehydration salts, which are just taking the oral rehydration therapy a little bit further, because there not only are we adding saline and glucose, but we're also adding back potassium as well as citrate or bicarbonate to help treat the acidosis and correct for the low potassium levels at the same time. And so they see, if we just talk about oral rehydration salts without understanding how that happens in the big picture, they don't really understand it. But when we present the case in the way where they actually see what happens with the loss of potassium and that they have metabolic acidosis, they understand why these oral rehydration salts work. And then we talk about how, what are some other drug targets that could be potentially developed for inflammatory diarrheas, and really just thinking about how you can target the transporters that we talked about to either prevent chloride secretion at the end or stimulate sodium absorption more at the beginning, if we think about it in that way. Okay, so key concepts that I think are important are that the intestinal epithelium plays a critical role in barrier function, as well as secretion and absorption. The intestine is able to handle a very large volume of fluids, which is driven by ion transport. And again, we only really covered a little bit about sodium and chloride, but contributions for potassium and calcium and magnesium are all really important as well. Any dysfunction in this can lead to electrolyte disturbances in the whole body. And so again, it's a chance to really show how the gut can integrate with all organ systems in an integrated way, which I think is great for particularly med students to understand. And then we use diarrheal disease as a great example to demonstrate these transport processes and pharmacological targets. And that's all I have for you today, so I'd be happy to take any questions or ideas from you. Thanks for your time.
I know you don't want to talk about the kidney, but I was curious about the hyperphosphatemia and the effects of tenapanor, for example, in the proximal tubule and how it impacts proximal tubular phosphate reabsorption, because that's, as you know, a major site of phosphate reabsorption, and parathyroid hormone
Right.
acts to inhibit it there.
Yeah, so the interesting thing about tenapanor is it's an oral NHE3 inhibitor, so it's really only blocking NHE3 in the intestine specifically. That seems to have some effect on phosphate uptake from the gut. It should not get into circulation.
Bob Speth, Nova Southeastern University. You were talking about the surface area. And one of the things I've come across is something called the Barker hypothesis. It's associated with epigenetic changes in the fetal condition with malnutrition. And it causes the development of what's called a thrifty phenotype. And I'm just wondering if anybody has ever looked to see if there's an increase in the surface area of the intestine associated with the development of this thrifty phenotype.
That's a great question. I'm not aware of anything off the top of my head, but there might be something out there. I just haven't looked into that particular hypothesis and if that affects that. But that would be really interesting to see if it changes the surface area.
Okay, thank you.